previously do it ahead of time to post it, so my bad, guys. But these questions are based on your lecture notes from class, and I'll be sure to go slow enough so you guys can follow along. This is going to be interactive, so I'm going to go over these questions, and we'll go through each answer choice one by one, pick apart what's true or false about these. I tried to make them in the style of how I remember her tests were during class. But other than that, we're going to end, I think this session goes from 12:30 to 2:30, so we're going to end 15 minutes early so that we have enough time to clean up and get the room ready for the next session that's going to come in after us. I think there's a next session at 2:30, or possibly a little after. But other than that, there's some evaluation forms that's on the front tables here, and also have a stack of them up here. Make sure you fill one out before you leave, and make sure you bubble in the session number, which is 84 for this session. It's really important that you bubble in 84. But other than that, we should have probably time left at the end for questions. There's around like 22 questions I have created from this. And yeah, so if you have any questions during the session, feel free to raise your hand, and we'll go over those as well. But for now, does anyone have any questions? Oh, you can take pictures. It's fine. Yeah, you're good. Other questions that I can see? Okay. Then we can start. So this first one says, I think it's about molecular biology's first portion. I think I grouped them into what chapters you guys were going over, so this is the last chapter, I think. So number one is, which of the following is true concerning transcription and translation, specifically in prokaryotes? So letter A, we have introns are removed and exons are joined to create mRNA. So for that answer choice, do we like that answer choice? Do we not like that answer choice? What is wrong with that answer choice? What is right about it? Okay. Yeah, so with introns and exons, those are going to be located in eukaryotes, not prokaryotes. So there's no mRNA splicing going on in prokaryotes. So then A is not right. So then we go with B. One mRNA strand codes for one polypeptide chain. And I heard a no. So what's wrong with that? Okay, yeah, so that's true. So usually in prokaryotes, one mRNA strand is going to code for two polypeptide strands. So that's why B is not correct. And then we have letter C, a methylated guanine nucleoside is added to the 5' end of the mRNA. Do we agree with that? Do we not agree with that? What sounds wrong? What sounds right? Okay, we don't think that's right. Okay, so that guanine nucleoside, I think it's also supposed to be nucleotide, that methylated guanine nucleotide is going to be added to the 5' end in mRNA in eukaryotes and not prokaryotes. So that 5' cap is for eukaryotes, just like the poly A tail is in eukaryotes. So none of these are related to prokaryotes, so the answer for this one is none of the above. Okay. Okay, so do we have questions on that one? Are we okay? Can we move on? We're good? Okay. So this was the answer. E was the answer, none of the above. Okay. So we have number 2. Which of the following is true about the structure of DNA? Oops, go back up a little bit. Which of the following is true about the structure of DNA? So A, we have it's composed of a polymer of deoxyribonucleotides held together by hydrogen bonds. Does that sound right? Or what could be wrong with that? So DNA structure. So we're looking at a strand of DNA. Does it contain deoxyribonucleotides, first of all? I see not. So yes, we have DNA, we have deoxyribonucleotides. Are those nucleotides held together with hydrogen bonds, though? The nucleotides themselves. So we're not talking about the bases in them, we're talking about the subunits holding those together. So like that sugar phosphate backbone, is that held together with hydrogen bonding? No, it's not. Okay. So hydrogen bonds are holding together the bases in between two strands of DNA, but they don't hold together the nucleotides in one strand of DNA. So A is not true. B is adenine forms between adenine forms two hydrogen bonds with 5-methyluracil in DNA. What does adenine usually form hydrogen bonds with? Adenine forms bond with guanine? Thymine, okay. So adenine forms bonds with thymine. What's the difference between thymine and uracil? One's in DNA, one's in RNA. What about the structure of thymine and uracil? How are they structurally different? Actually, both of them are going to be the pyrimidines. So we can't go with that either. Something with the methyl group. So actually, another name for thymine is this 5-methyluracil because it has a methyl group on the 5 carbon in the ring. So technically, B is correct. Adenine does form two hydrogen bonds with 5-methyluracil because 5-methyluracil is thymine. Okay. So far, A wasn't correct because we don't have hydrogen bonds between the DNA nucleotides. And B was correct because 5-methyluracil is the same thing as thymine, and A, hydrogen bonds with T, and they form two hydrogen bonds. But we're not done yet because there is an answer choice for B and C, so we'll see what C is. C says the sugar phosphate backbone is held together by phosphoester bonds. Okay, so that sugar phosphate backbone is actually held together with phosphodiester bonds, so it forms two esters on each side of that phosphate group, or with that phosphate group. So C is also wrong, so D and E are wrong because A and C are wrong. So our answer choice that we're going to pick, because we want to know what's true, is going to be B. Adenine forms two hydrogen bonds with 5-methyluracil, because that's another name for thymine. Okay. Do we have questions on DNA structure? Yes. The other ones probably do have different names, but not necessarily in terms of the other nucleotides. Thymine was just a special one because it was the same structure as uracil, just that one methyl group difference. But if you look at the structure of the other bases, there's more than just one difference on them. So you'll have to change a bunch of things about them. But I think this was one of the more highlighted ones if she's talked about it in class. Okay. Other questions? Okay. So then number 3 is if DNA is labeled with nitrogen, with light nitrogen, so N14, and then the cells are allowed to replicate in heavy nitrogen, N15, for 2 generations. So we have the beginning with just 2 strands of light nitrogen DNA. We let those replicate in heavy nitrogen, and that's going to occur twice. So it's going to replicate. We're going to have 4 sets of, 4 sets of DNA at the end. So we want to know, after we isolate the DNA and put it in the ultracentrifuge, we want to know what's going to be the most likely occurrence, or what the centrifuge cell is going to look like at the end. So where are our bands going to show? Are we going to have 3 bands, or are we going to have 2 bands? Are we going to have only light, are we going to have light, heavy, or hybrid going on with this? So the first question pretty much is, how does DNA replicate? When we replicate this light nitrogen DNA, is that going to stay the same? Are we going to have light nitrogen with light nitrogen and then heavy with heavy in that first generation? Or are we going to have hybrids in the first generation? Is that first question. Maybe let me reword that probably. So DNA replication, what happens first during DNA replication? Wait, can you speak louder? Okay, so we split the 2 strands apart, And then we're going to replicate them separately. So it's semi-conservative in that regard. So we're going to split them up, and then we're going to replicate them. So we're splitting up this light nitrogen DNA. So we have one light strand, and then we're going to replicate that. The only nitrogen that's available to replicate that is the heavy nitrogen. So we're going to have the light nitrogen splitting up, and they're going to replicate with heavy DNA. So we're going to have two hybrids in the first generation. But it's asking for the second generation. So then what's going to happen in that second generation? We're going to have that DNA, that hybrid DNA, it's going to split again. And now it still only has heavy nitrogen to replicate with. So we're going to have another hybrid, and then we're going to have heavy and heavy together. And that's going to happen for both of those hybrids that you split apart in the first generation. So our answer choices are A, there will be 3 bands, 1 light, 1 hybrid, and 1 heavy in that second generation. So does that make sense? Would we have light and light staying together to make that light band? No, okay. So because light nitrogen is what we started with, and DNA replication is semi-conservative, we shouldn't see a light band. We shouldn't have the light nitrogen DNA strands staying together and creating that band. So anything with light should be kind of taken off. So we have light, light, and light. So that kind of gets rid of 3 answer choices right there. We also have none of the above answer choices down here. So if we look at that, we said light is the beginning, and we can't have any light in the end because DNA is semi-conservative when it's replicated. Our answer choice then would be, there will be 2 bands. One would be hybrid, and one would be heavy. Or none of the above. So do we agree that we'll have a hybrid brand and a heavy brand? Yeah. So in that second generation, we're going to have DNA strands, or double-stranded DNA strands, that only have heavy nitrogen, and we're going to have DNA strands that have a mixture of heavy nitrogen and light nitrogen. So the answer choice for number 3 should be D. Four bands. The DNA strands, I think, are sticking together. So what do you mean there'd be 4 bands? So there'd be 4 sets of strands, or 4 DNA pieces, but 2 and 2 are going to have the same mass because 2 sets are hybrid and 2 sets are heavy. But there's going to be 1 band for each of those. Yes. Draw it? Yes. Try drawing it over here. If you guys can see this board. So I'll draw it up here maybe. I'll erase this portion. Okay, so the light is going to be in green and the dark will be in blue. So at first you have 1 or 2 light strands together. Then DNA is going to replicate. So that means these light strands are going to break apart. Okay, so these light strands are breaking apart. This is the first generation. And they're going to replicate in heavy DNA. Or not heavy DNA, sorry, in heavy nitrogen. So that means the nitrogen on the bases are going to be that darker one, or the heavier one. So this is going to be in blue. So then they replicate with this heavy nitrogen. So in this first generation, this first replication, we have hybrids. Okay. Then in our second generation, these 2 strands, or these 2 sets of strands, are going to break apart again. So then we're going to have a light strand, another light strand, a heavy strand, and another heavy strand. Maybe I'll move this over. Heavy strand. And they're going to be split apart. And then those are going to replicate also in heavy nitrogen. So they're going to have, they're going to be replicated in heavy nitrogen, so then we're going to have a hybrid. We're going to have just a double-stranded DNA with just heavy, another hybrid, and another double-stranded DNA with just heavy. So in the centrifuge, these two heavies, or these two double-stranded DNA segments with the heavy nitrogen are going to fall in the same band because they're both the same mass. And these two with the hybrid portion of nitrogens, they're going to form their own band as well because they have a different mass, because they have both heavy nitrogen and light nitrogen. Okay. Does that help clear it up a little bit more? Okay. Do we have other questions on, I guess this was the, if I remember correctly, that experiment was the Hershey Chase. But this is based on one of the experiments that they had to show that DNA was semi-conservative. Okay. So do we have other questions on DNA replication or DNA structure? Are we okay? Can we move on? Okay. So then our answer choice for this, because we don't have a light chain, we only have hybrid, hybrid, heavy, and heavy, we're going to have one hybrid band and one heavy band. So our answer choice is D. Okay. So then we have number 4. Oh, sorry. We have number 4. During DNA replication, which of the following we're going to figure out, I guess, which is true. So A was DNA polymerase reads the template strand of DNA in the 5' to 3' direction. Okay. So do you agree with that statement? Do we disagree with that statement? What could be wrong? So DNA polymerase, we want to know the directions that DNA polymerase reads the template strand and what directions it actually replicates DNA or synthesizes the DNA. So what are those directions? Is it 5', 3', or 3', 5'? So is that the direction for reading the template or for synthesizing DNA? Okay, so this 5' to 3' is the direction for synthesizing DNA. So it starts synthesizing from 5' to 3'. So it adds on to the 3' end, going from 5' to 3' when it replicates. But it's going to read the other strand in the opposite direction. So it's reading the 3' end of the template strand, so it can begin its 5' strand, because DNA is anti-parallel. So the 3' of the template strand is going to match up with the 5' of the new strand that's being made. So that new strand is made in the 5' to 3' direction. The template strand isn't read in the 5' to 3' direction. It has to be read in the opposite direction of DNA replication. So whenever you hear 5' to 3', that's for the synthesis of DNA. That's for the replication of DNA. Okay. So during DNA replication, the DNA polymerase is going to read the template strand 3 to 5 so that it can synthesize 5 to 3. So A is not correct. B is primers are synthesized by DNA polymerase 1. Is that the enzyme that creates primers? No, what's the enzyme that creates primers? It's primase, so yeah, which kind of goes better. So not DNA polymerase, it's going to be primase that creates that primer. So that's also not true. C is primers on the lagging strand are removed by DNA polymerase 3. Okay. So what's going to remove the primer on the lagging strand, or for each of those fragments to be replicated? So what enzyme is going to do that? Okay. So instead of DNA polymerase 3, it's going to be DNA polymerase 1. That's going to simultaneously remove those primers and replicate DNA. And then what goes in and attaches those fragments together after DNA polymerase 1 takes out the primer and replicates DNA? There's going to be a little nick in it. Something has to, so ligase is going to glue those together after DNA polymerase 1 removes the primer and replicates DNA a little further. So C is also not correct. So the answer is choice D, which kind of looks like it has to be right, but we'll go through it and see that is right. So DNA helicase separates the two strands of DNA, and then DNA gyrase removes that supercoiling when you separate the two strands. because in prokaryotes, what structure is DNA in? So it's double-stranded DNA, but is it in a strand or is it in a circle? Yeah, so it's going to be in a circular DNA strand. So when those strands in that circle are pulled apart, there's going to create that supercoiling effect. So DNA gyrase is going to remove that supercoiling effect. Okay. So our answer choice for during DNA replication, we're going to have these enzymes working. So it's our answer choice for number 4 is going to be D. Do we have questions on DNA replication and the enzymes that are used during it? Possibly? Okay. We can always save them for later as well. So then number 5, we have during transcription. So we're going to figure out what's going to happen during transcription. So letter A is the core enzyme of RNA polymerase binds to the promoter region on the DNA template. So we're trying to look at, okay, what region of RNA polymerase binds to what region of the DNA template. So we're saying the core enzyme binds to the promoter. So does that sound right? Does that sound wrong? What part of RNA polymerase binds to the promoter? It's the sigma portion. So the core enzyme is the outer portion. The sigma portion is what's going to bind to the promoter. So A is technically not correct. So there's 2 portions that you want to take note of. For RNA polymerase, there's the core enzyme portion, what's going to do most of the actual transcribing, and then there's that sigma portion. The sigma portion is going to bind to that promoter during initiation. Okay. So this is not correct. It should say the sigma portion of RNA polymerase to be correct. Then we have letter B. Ribonucleotides are added to the 5' end of the transcript when we're creating mRNA or the RNA. Okay. So how is the so it's similar to DNA replication, but we're using RNA polymerase and we're creating ribonucleotides versus deoxyribonucleotides, but the directions are the same. So what direction would the RNA be synthesized in? Is it 5' 3' or 3' 5'? It's going to be that 5' to 3' direction. It's going to be synthesized in the 5' to 3', just like in DNA. So if we add nucleotides to the 5' end, what direction would that be going? Would that be going 5' to 3' or 3' to 5'? That would be going the opposite direction if we added to the 5' end. We'd be starting at 3' and we keep adding to 5'. We get longer that way. That'd be 3' to 5'. But RNA polymerase synthesizes RNA in the 5' to 3' direction, not the reverse. So in order for this B to be true, ribonucleotides would have to be added to the 3' end of the transcript. So the transcript is what's being transcribed. The transcript isn't going to be that template DNA that we're talking about. Okay. So that kind of also ties in letter C. So transcription occurs in the 5' to 3' direction. Was that true? I see confused faces. Okay. So transcription and DNA replication are both going to occur in the 5' to 3' direction. That's how the strands are being made, 5' to 3'. That template strand is maybe I'll draw a little bit, and that might help a little bit. I don't know. I'll erase this portion. Okay, so if we have 3', 5', and this is our template DNA strand. So if our template has a 3' on this side, 5' on this side, for RNA synthesis and for DNA replication, so for RNA synthesis, like this problem is that RNA polymerase is going to attach its promoter to this 3' end. It's going to attach to the 3' end. and it's going to add ribonucleotides to this. So it attaches to the 3' end, and it's going to add nucleotides. So it attaches to the 3' end of the template strand. And as it makes more and more, it creates more and more nucleotides that come on and on. This is going to be in that 5' to 3' direction. So this new strand is going to be 5' to 3'. So every nucleotide that's being added on is being added to the 3' end of the transcript. Okay. Does that make a little more sense, or? Okay. Okay. So then for number 5, A was wrong because it's not the core enzyme portion of RNA polymerase, it's the sigma portion. B was wrong because ribonucleotides have to be added to the 3' end of the transcript. So that C is correct. We transcribe DNA 5' to 3'. But we'll also, I guess, look at letter D as well, even though C is the correct answer. So letter D was the stem loop structure downstream from a run of uracils terminates transcription. So how would that, we know that's wrong, but how would that statement be turned true? Yeah, so instead of being downstream of a bunch of uracils, it's going to be upstream of it. So it's going to occur before uracils. Okay. And then, of course, not all of the above is taken out as well. So our answer choice for number 5 is going to be D. Not D, sorry, C. Transcription occurs 5' to 3'. Okay. Do we have questions on transcription? Or are we okay? Okay. So then number 6 talks about translation. So letter A is during elongation of translation, the growing polypeptide chain is attached to that tRNA in the P site of the ribosome. So this is pulling together a whole bunch of information in one answer choice. So what are the different sites in the ribosome? We'll start with that. So there's 3 different sites that this tRNA can attach to. So there's E, P, and A. So the exit, the attachment, and where that polypeptide chain will be is in the P portion, the polymerization portion. So we have E, P, and A. So during elongation, our growing polypeptide chain is going to be attached to that tRNA in the P site. So it's not going to be an exit site or the attachment site, because that's where either new tRNA are joining or the old ones that already, like, their amino acids that were attached to them already are part of the polypeptide chain are breaking off. So A is going to be true for that. But we'll go through the rest of the ones to see why those aren't. So B is the codon region of tRNA interacts with the anticodon region of mRNA. So which molecule has the codon and which molecule has the anticodon? Okay. So mRNA is composed of a bunch of codons, and what are codons made of? What do we call a codon? Okay, so 3 of those bases, so 3 of those ribonucleotides together are going to make that codon. And so that's part of the mRNA. So mRNA is going to be broken up into groups of 3, and we call those codons. And then tRNA is going to be that structure with the amino acid attached to it on one end, and it's going to have a region of that anticodon, or that complementary codon of the mRNA to match it up. So we can make a polypeptide chain of amino acids in the order that mRNA is instructing. So the codon region should be on mRNA, and this anticodon region should be on tRNA during translation. Letter C is there are a total of 3 different start codons. So what's wrong with that? Like, how many start codons are there usually? There's 1, and what amino acid does that usually code for? Remember that? No? Okay. It's usually methionine. And So there's only usually one start codon, but there's 3 different stop codons. Is kind of what that concept is confusing. So there's only one start codon. It usually codes for like the M amino acid or methionine, and there's going to be 3 different stop codons usually. But I don't think you have to worry about which ones are what. Like you don't have to remember the codon codes for it, I don't think. That might be later on if you go into microbiology. So then D is during initiation, the first tRNA hydrogen bonds with the start codon in the A site of the ribosome. So we know that's not right. So where does that first tRNA attach to the mRNA in the ribosome? Is it the E site, the P site, or I guess it's not the A site, we just figured out. It's going to be in the P site. So that first one is going to attach at the P site, but all other tRNA molecules are going to start in the A site, or the attachment site. So during translation, the answer choice was A, so it's not none of the above. So during translation, the elongation of the growing polypeptide chain is attached to the tRNA in that P site of the ribosome. That one was the correct one. Do we have questions on translation? Or the parts of the ribosome, or tRNA and mRNA? Okay. Then we'll go to the next one. Okay, so number 7 is, which of the following is true about viruses? So letter A, we have the viral genome can contain DNA or RNA. So is that true? Can we have DNA viruses and RNA or RNA viruses? So that's like what their genome is made of. That's what their genetic material is. Yes, so we can have DNA or we can have RNA in viruses. It doesn't matter which one. So then B is the viral genome can be single-stranded or double-stranded. So can we have single-stranded DNA or double-stranded DNA, or single-stranded RNA, double-stranded RNA? Yes, all of those answer choices are very possible in viruses. So we have answer choice A and B that's possible, but E, all the above is possible, so let's look at C. So a naked virus does not have a capsid, so we're talking about either the envelope viruses and the naked viruses. So do all viruses have capsids? Yes, so what is a capsid? Okay, so a capsid is that protein coat, so all viruses are going to have that protein coat, bless you, but the encapsulated viruses are going to have another coat on top of that protein coat to differentiate them from the naked virus. So the naked virus just has that genetic material, either double or single-stranded RNA or DNA with that capsid. An enveloped virus will have the same thing, but with an extra layer on the outside, what they call the envelope. Okay. So do we have questions on viruses? I think there was like 2 slides on viruses in this portion, but okay. Then we'll go to the next one. That was pretty straightforward. Number 8 is which of the following is true about bacteriophages, so specifically ones that go with bacteria. A is during the lytic cycle, the host cell is lysed. So that little lytic cycle, so we have lytic versus lysogenic cycle. So what's happening during that lytic cycle? Is this true? Is the cell lysed? What is the virus replicating, or is it inserting its genetic information into the host genome? What's going on? Okay, so that cell is going to lyse. So during the lytic cycle, the virus is going to, or the bacteriophage in this case, is going to replicate, and it's going to lyse that host cell. And then in contrast, during the lysogenic cycle, the viral genetic information is going to be integrated into the host genome. So that's the letter B. Do we agree with that one, or does that sound wrong in a way? Okay. So I hear no objections. So during the lysogenic cycle, instead of replicating the virus and lysing the cell like in the lytic cycle, it's going to incorporate its genetic information into the host genome, and then it's going to be replicated with the host cell. Okay, and then we'll look at C as well. So a prophage in the lysogenic cycle can be induced to undergo a lytic cycle and lyse the cell. So we're saying after this bacteriophage went through the lysogenic cycle and incorporated its genetic information into that host genome, it can then come back and go through a lytic cycle and kill the cell. Does that sound right or not? So once it's in the lysogenic cycle, it can go back into the lytic cycle. So the answer choice for A would be that all of the above. Okay. Do we have questions on lytic cycle or lysogenic cycle? Okay. Well. So number 9 is a retrovirus. We're talking about retroviruses. So A is, it's composed of double-stranded RNA. So the first question is it RNA or DNA in a retrovirus? Okay. So in a retrovirus, it's going to be that RNA because it's going to take that RNA, it's going to convert it back to DNA. And that's kind of the hallmark of a retrovirus. But then the other question is it double-stranded RNA or is it single-stranded RNA? Yeah. So for this retrovirus, or for retroviruses in general, it should be single-stranded RNA. There's 2 single-stranded RNAs in a retrovirus. So A is not correct for that because it says double-stranded. So it should be single-stranded. Then B, a retrovirus has a reverse transcriptase enzyme that converts that single-stranded RNA into DNA. So that one is true because that's kind of the definition of a retrovirus. It's going to have RNA, and then it's going to have that reverse transcriptase enzyme to convert RNA back to DNA. Then C is that double-stranded DNA is integrated into the host cell and integrated into the genome specifically of that host cell. So after reverse transcriptase converts its single-stranded RNA to DNA, then that DNA is going to synthesize the other strand and then integrate into the host genome. So do we agree with that one, or does that not sound right? Okay. Okay. So that sounds right. So if the host cell has double-stranded DNA, then this retrovirus is going to have to create double-stranded DNA in order to get its genetic information into that host cell. So for number 9, both B and C are correct. So the answer choice for this one is E. A wasn't correct because it's double-stranded and it should be single-stranded RNA in the retrovirus. Okay. I'll move this down a little bit. Okay. So do we have questions on retroviruses or okay? Or reverse transcriptase or single-stranded, double-stranded DNA or RNA? So number 10 is, which of the following is not a mechanism for gene transfer among bacteria? So looking for non-mechanisms. So for A, we have restricted transduction. So does anyone know what restricted transduction is? Or what's going on during that? Is that a form of gene transfer? Get so many confused faces. Okay, restricted transduction is a form of gene transfer. I think she goes over it a lot, like more in your notes. There's even a picture of the process as well. If I remember correctly, that's the one where a cell is going to, or one bacteria is going to, restricted transduction. So a virus is going to, no, it's either the one where a cell is lysed and its genetic information is taken up by another cell, or it's the one where a virus is going to lyse a cell after it's been through the lysogenic cycle. So after it's already incorporated its DNA into the host cell, and after it takes its DNA back out, it's going to take some of the host cell with it and infect another cell with it. I don't remember which one's which, but it is a form of gene transfer in the case of this question. So that might be something you want to look over in your notes. It's in the molecular biology portion of it. Then B is conjugation. So is that a form of gene transfer among bacteria? Okay, so conjugation is, does anyone know what conjugation is? Like what's going on during that? What type of genes are being transferred there? So there has to be, yes, there's contact with each other, yeah. So during conjugation, I think there's actually two forms. One form is with that plasmid. So one cell is plasmid positive. It's going to copy its plasmid and then transfer it over to the other one. So that one could be F positive as well. So conjugation can either be the F positive and F negative to both F positive, or it can have that HFR factor that it transfers over, which is when the plasmid is integrated into the cell's genome itself. So the cell doesn't just transfer the plasmid over, it also transfers some of its own genome to the other cell. So those are like two different forms of conjugation that she kind of goes over in the chapter. Okay, and then letter C is transformation. Okay, then I'll give that one away. So that is another form of gene transfer. I think that one might be the one that's referring to the viral transfer of them. It was either restricted transduction or transformation with those. I'm not sure exactly which one, but they're all forms of gene transfer. It's not just A or C. It's going to be, so we're looking for what's not a mechanism. So all three of these are a mechanism. So the answer choice for this one is going to be E. Number 10 is going to be E. Because restricted transduction, along with general transduction, the two forms of conjugation and transformation are all gene transfers between bacteria. So do we have questions on gene transfer? I think those are just the main ones that she goes over. I don't know if she goes over any more. I think that's the portion of the microbiology stuff, and then the next portion I think is human microbe interaction stuff. So we can go over that. We don't have questions now? Okay. So the number 11 is going to focus on diphtheria. So we're looking for the answer choice that is false for this one. So our answer choices are A, it's going to inhibit transcription, so that protein synthesis cannot occur. Okay, so we want to look back into what diphtheria does. We're not sure about that one, it looks like. So we'll move on to letter B then. So B is, diphtheria is an AB toxin. So that means it has an binding portion and then the actual antigen portion. So what do we remember about diphtheria? Anyone? No? Okay. So let's look at the third one then. So then AB of diphtheria is a cytotoxin. Another one. So what is a cytotoxin then? What does it sound like? So it's toxic to the cell. So it kills cells. So it leads to cell death. It's not very specific, but it's just going to lead to cell death. So AB toxins are usually cytotoxic. Then we have letter D, is it's found in Coronium bacterium of diphtheria during the lysogenic pathway induced by phage beta. So it seems no one knows these. Okay, so diphtheria is an AB toxin, and it's also a cytotoxin. So it has two portions of it, and a binding portion and an antigen portion. The antigen portion is going to lead to cell death. And this AB toxin diphtheria does come from this bacterium. And it only comes from this bacterium after phage beta, this virus, inserts its own genetic information through the lysogenic pathway into that bacterium. So this bacterium by itself is not toxic itself. It needs to be infected with this phage to be toxic. But it is an AB toxin and it's cytotoxic, so these three are right. The problem is with this A, with letter A. So it's not going to inhibit transcription, so that protein synthesis doesn't occur. So because transcription, is that from DNA to RNA, or is that from RNA to protein? What's transcription? We went over this earlier, so you guys should know this, because no one asked questions. So transcription, so is that DNA to RNA or RNA to protein? So transcription is DNA to RNA, and for diphtheria, that's not going to mess up the DNA to RNA. It's going to actually mess up in translation. So adding the proteins on to make that polypeptide chain. So it disrupts translation, not transcription, and that's going to result in the cell death, because if the cell can't form proteins, then it can't survive. So when we're looking for what's false, that'll be letter A. It's not going to inhibit transcription, it's going to inhibit translation in cells. Yeah, but diphtheria is an AV toxin and is cytotoxic, and it does come from that bacterium. Okay. So then the next one is, which of the following is true about hyaluronate? I can't say it. So this enzyme right here, it ends in ACE, so it's an enzyme. So we want to know what is true about the enzyme. So the first two answer choices are where this enzyme will be located. So is it going to be found in E. coli or Staph aureus, or both, partially, because we have an answer choice for both. And then C is that this enzyme is a spreading factor that allows bacteria to adhere to a host. So we're saying that this is a spreading factor and it adheres to a host. So does anyone remember what this enzyme does, where it's found, possibly? Yeah, so this is to help break through the basement membrane, and it actually works with not for adherence, but for invasion of the actual host cell itself. So it's not going to be a spreading factor for adherence. It's going to work not just in adherence, it's going to work after the bacteria has adhered to the host. It's going to work with invasion. So C isn't correct. And then this enzyme is found in 3 different types of bacteria. It's found in, I think it was in Staph, Strep, and Clostridium. So it's not going to be found, or not going to be secreted by E. coli, but it is going to be secreted by this Staphylococcus aureus. So for number 12, B is going to be correct. So it's not found in E. coli. It's only found in Staph, Strep, and Clostridium. And then C, it's not going to be a spreading factor for adherence. It's going to work with invasion of the host cell itself. Okay, so the answer choice was B. Okay. Do we have questions on, I guess, diphtheria or this enzyme or anything of that sort? Or adherence or invasion? Answer to 12. So answer to 12 was B, Staph aureus, because that's where it's found. So this enzyme is found in Staph, Strep, and Clostridium. It's not found in E. coli, so it's not going to be secreted by E. coli. And then this enzyme isn't a spreading factor for adherence. It's going to work after the bacteria has already adhered to the host cell, and it's going to work in invasion, not host cell, but it's going to work with invasion, breaking into the basement membrane and causing damage to the cell, not to sticking to the cell first before it can cause damage. So this is actually causing damage. Okay. So, other questions? Mm-hmm. Yeah, so it's going to inhibit translation, yeah, not transcription. So it's going to inhibit that elongation of that polypeptide chain. It's not going to inhibit creating that mRNA. Okay. So then number 13, we have this scenario. So a person is going to ingest food that was improperly canned, and they're going to develop botulism, and that's excess information we don't need to know. So then we want to know which of the following is most likely going to be true about this botulism. So A is that person has an endotoxin. So is the botulinum toxin, is that an endotoxin or an exotoxin? Okay, so that's an exotoxin. So that's not an endotoxin. Where are endotoxins found? Are those in gram-negative or gram-positive bacteria? Yeah, so endotoxins in gram-negative, it's going to be in that LPS layer. this botulinum toxin is coming from not a gram-negative bacteria. So it's not an endotoxin, it's going to be an exotoxin. So then B is this person has an enterotoxin. So where do enterotoxins act on? What part of the body? So it's like the digestive system, enterotoxin. Does botulinum toxin, does that affect the digestive system, or what does that affect? Okay, so that affects like muscles and stuff. So that's going to be a neurotoxin. It's going to affect the nervous system or that neuromuscular junction. So instead of an enterotoxin, it's going to be a neurotoxin. And instead of an endotoxin, it's an exotoxin. So A and B are not right. And then for C, so for C and D, we have two different scenarios. So either the person's going to have flaccid paralysis due to blocking of glycine channels. So what do glycine channels do in that neuromuscular junction? Do they stop contraction or not stop contraction? Or is this too much? Yeah, this is probably too much. Anyway, there's a picture that shows what this botulinum toxin does in her slides. So it's not going to block glycine channels because that wouldn't actually make flaccid paralysis because glycine stops contraction. So for D, this person has flaccid paralysis due to blocking of acetylcholine receptors on the postsynaptic terminal. So what does botulinum toxin do in that neuromuscular junction? Okay, so instead of blocking the receptors on that postsynaptic terminal, it's going to block the release of acetylcholine itself. So acetylcholine is just not going to exit the nerves themselves. So A through D is wrong, and our answer choice is none of the above. Because the person's going to have flaccid paralysis, but it's going to be due to blocking of acetylcholine receptors on the presynaptic terminal or blocking the release of acetylcholine. Okay, so that answer choice is E. Do we have questions on botulinum toxin, or are we okay? We're okay. Move on. Okay. Number 14 talks about cholera enterotoxin. So A is that it's an AB toxin that binds to this GM2 ganglioside on intestinal epithelium. So I know, okay, so it binds to ganglion cells. So is it binding to GM2, or which ganglioside is it binding to in intestinal epithelium? So that B portion of that AB toxin, where is that binding? Okay, so instead of GM2, it's going to be binding in GM1. So that's what makes this wrong. So in order for this to be correct, it should be an AB toxin binds to GM1 on the intestinal epithelium. Then we have B. This cholera is going to cause chloride and bicarbonate to be secreted into the lumen of the intestines, and that's going to result in water retention. So does that sound right? Or, yeah, so that's not exactly right. So when these ions are being secreted into the lumen, water is going to follow them. And when it's secreted into the lumen of the intestines, that means we're going to have water loss. An enzyme that's going to convert ATP to CH. Does convert ATP to CAMP or increases CAMP. But these other two answer choices were wrong, so number 14 is going to be C. This cholera enterotoxin is going to activate adenylyl cyclase, which is the enzyme that converts ATP to CAMP. A was wrong because it says GM2, but it should be GM1. And then B was wrong because instead of water retention, we're going to have water loss, because water is going to follow chloride and bicarbonate into the lumen of the intestine. Okay. Do we have questions on that? Okay. So then number 15, number 15, if the LD50 of bacteria A is 55 cells, and the LD50 of bacteria B is 25 cells, we want to know which of the following is going to be true for that. So letter A, we're saying that, or before we look at the answer choices, what does LD50 tell us? What is that measurement? So lethal dose 50. So the lethal dose to kill off 50% of a sample. So if it has a high lethal dose, does that mean it's very toxic, or does that mean it's not very toxic? If it has a very high lethal dose, not very toxic. So the more you need, or the higher your LD50 is, the less toxic your substance is. So because bacteria A has a higher LD50 than bacteria B, bacteria A is going to be less toxic than B so far. So our answer choice was bacteria A is more lethal than B, so that's like the opposite wording of what I said. So A is more lethal than B. So A has a higher LD50, so it's not going to be more lethal than B. It's going to take less of B to kill off 50% of a sample. So letter B says bacteria B is more lethal than A. So according to these LD50s, B would be more lethal than A in terms of like the same dosage. So A is not correct, but B is correct. But there's an answer choice for B and C, so we'll move on. So letter C says it would take 25 cells of bacteria B, because bacteria B has a LD50 is 25. So it's going to take 25 cells of bacteria B to kill 50 animals. Is that what LD50 means? No. So it's not going to necessarily kill 50 animals in the sample if we're like testing on animals in this case. If it's out of 100, then that would technically be right, because that would be 50%, and LD50 is for 50%. But it doesn't say that, it just says 50 in general. So C is not correct, but B is. So our answer choice was B, because the lower the lethal dose, the more lethal something is. And B has a lower lethal dose than A. Okay. So then number 16 is going to talk about natural killer cells. So this is going into immunology portion. So do we have any questions on human microbe interaction? I think that was all the questions I had on that one. Or we look okay? Okay. So number 16 talks about natural killer cells, and we're going to see how they're classified. This is a check all that applies, so we can have multiple answers, or we can just have one answer. Either one goes. So for this one, A, so natural killer cells, are they a part of innate immunity, or are they specific immunity? Or are they non-specific? So innate immunity, so that's, what would constitute innate immunity? What does innate mean? Yeah, so innate is going to be non-specific, so these are actually the same thing. So natural killer cells, do they only attack certain things with memory, or is it just for any infected cell? Okay, so these natural killer cells are going to be part of this innate immunity. They're going to infect, or not infect, they're going to kill off infected cells, like cancer cells, for example. So they are a part of innate immunity, so A is one of the answers. Because innate immunity and nonspecific immunity are the same thing, D is also an answer. Then B says natural killer cells are antibody-secreting plasma cells. So is that true? As false. So what kind of cells turn into antibody-secreting plasma cells? Those are B cells. So B is not correct for this one, because this is talking about B cells, not natural killer cells. C, natural killer cells are part of cell-mediated immunity. Okay, so are natural killer cells cells? Okay. And they're going to induce cell death about infected cells. So that actually meets the criteria for cell-mediated immunity. So they are not only part of innate immunity, which is also specific immunity, they're also part of cell-mediated immunity, these natural killer cells. Okay, so then what else? So then letter E says that these natural killer cells are lymphocytes. Or lymphoid cells is another way of saying that. Is that true? What kind of cells are these? Okay. They actually are lymphocytes. or a type of lymphocyte, or a type of lymphoid cell, same thing. So for number 16, natural killer cells are part of innate immunity, so that's letter A. They're part of cell-mediated immunity, that's letter C. They're part of nonspecific immunity, that's letter D. And they're lymphocytes, that's letter E. So it's all the letters except for B, because antibody-secreting plasma cells are B cells, not natural killer cells. Do we have questions on natural killer cells, or maybe any questions on innate immunity, nonspecific immunity, or lymphocytes, or, I mean, there's more questions that go over this as well. Okay, no hands. Yeah. Why is C correct? It's part of cell-mediated immunity. So cell-mediated immunity, so certain cells are going to either phagocytize or induce cell death on other cells or other infected cells. So these natural killer cells are cells, and they're going to go around and find affected cells and going to induce programmed cell death on those. So that's why it's part of cell-mediated. Also, because it's in your notes. That's the other way. I think she lists certain ones that are part of cell-mediated immunity, and natural killer cells were part of the list. Other questions? Okay. So then, number 17, which of the following is true about B cells? So letter A, we have it contains MHC class II proteins on the cell surface. Okay, so what kind of MHC proteins do B cells have on their cell surface? What kind do they express? Are they class 1 or class 2? 50-50 chance. Okay. They're going to have, this is true, they do have these class 2 proteins on their cell surfaces. It's these B cells. They're going to do that. There's a few other ones. But all cells, or all nucleated cells, are going to have class 1. But B cells are one of the special ones that have class 2. Okay. And then letter B, since we have more answer choices that go with letter A, we're going to look at letter B and C. So B says that B cells are going to differentiate into antibody-secreting plasma cells. And from that last question, we figured out that that was true. This is going to be for B cells, so letter A and B so far are correct, and we have an answer choice for A and B, so that one will be our answer. But let's see why C is not correct. So C says B cells produce cytokines that increase phagocytosis in macrophages. So if B cells don't do that, what kind of cell does that? Okay, so helper T cells. Do you remember, like, specifically if they're helper 1 or helper 2 that does that? Helper 2? It might have been helper 2. Yeah, I think helper 2 is the one that induces B cells to become these to differentiate into antibody-secreting plasma cells. And then T helper 1, I think, are the ones that are going to release those cytokines for macrophages. And there's also TC cells that are going to produce cytokines, but not necessarily from the macrophages. Okay. So do we have questions on B cells? Okay. So then number 18, which of the following is not a mechanism that bacteria use to inhibit phagocytosis, so they don't want to be engulfed and broken down? So which one is not a mechanism? So there's probably a bunch of mechanisms. We don't know which one's not. So A, we have the production of carotenoids that neutralize singlet oxygen. So singlet oxygen is something that a phagocytic cell would use to break down that bacteria. So would this be a good way to inhibit phagocytosis? Yes. So this is a way. I think it might have been either Staph aureus or Strep pyogenes that did this. I don't know if you need to remember exactly what microorganism does what. But this is a way that bacteria or certain kinds of bacteria can inhibit phagocytosis. So this one is a mechanism. We're looking for what's not. Letter B says PAMPs interacting with PRRs. So what does PAMP stand for? Okay, so these PAMPs are going to be on the pathogen itself. Pathogen activating. Okay, never mind. I don't remember what it stands for either. But I think the PRRs are going to be on the phagocytic cells themselves, and the PAMPs are coming from the actual pathogen. So when PRRs interact with PAMPs, that's actually going to induce phagocytosis. That's going to speed it up instead of stopping it. So it's going to have the opposite effect. But you also want to take note of what they stand for. I did not have that in my notes up here, and I don't remember off the top of my head. My bad. But that's something to look in your notes for, because she could just write the whole word out anyway. So that's a mechanism that is not a mechanism to stop phagocytosis. That actually causes it to happen more readily. So this B would be an answer choice, or would be the answer choice for number 18. So bacteria do contain capsules to stop phagocytosis, and some of them produce leukocidins. So that's, I guess, against being broken up by the leukocytes. So leukocidins and bacterial capsules and these singlet oxygens, or carotenoids that neutralize singlet oxygen, those are all mechanisms to inhibit phagocytosis. But interactions between PAMPs and PRRs actually causes phagocytosis. It doesn't inhibit it. Okay. So then we have number 19. Which of the following is false about T cells? So instead of B cells, we're talking about T cells now. So letter A is that macrophages act as antigen-presenting cells to the Th1 cells. Is that one true? So we're looking for what's false. So macrophages act as antigen-presenting cells to Th1 cells. So we're going to have macrophages. They're going to have the MHC complex with the antigen attached to it. It's going to present to these Th1 cells, and these Th1 cells are going to release those cytokines to cause macrophages to induce phagocytosis. So letter A is true about T cells, or about Th1 cells specifically. So we're looking for what's false. So A is true. Letter B says that Th2 cells act as antigen-presenting cells to B cells. That's false. So how would this be true then? How would we fix this sentence? So Th2 cells are not antigen-presenting cells. What are antigen-presenting cells in terms of Th2 and B cell interaction? Okay, because we know that Th2 cells are going to activate our B cells, because we talked about that, I think, prior. Actually, that's letter C. So Th2 cells release cytokines in order to activate B cells. And before we said that Th2 cells do activate B cells, and that activation of B cells causes them to differentiate in those plasma, the antibody-producing plasma cells. So C is correct. It's going to release cytokines to activate B cells to produce antibodies. But B is not correct. B is the false one, because Th2 cells are not antigen presenting to B cells. It's actually the opposite way around. So B cells with the MHC, I think complex 2 complex, that's going to present antigens to Th2 cells, and then Th2 cells are going to then activate the B cells to produce antibodies. So B cells are going to be antigen presenting, not Th2, is why letter B is wrong. Okay. So it's true that macrophages act as antigen-presenting cells to Th1. B cells actually act as antigen-presenting cells to Th2. So that's why B is not correct. B is the answer choice for number 19. And then for C, Th2 cells do release cytokines to activate B cells. So that's how they activate them. Okay. So do we have questions on T cells or? Thank you. Okay. So then we have number 20. So which of the following is true about IgG? So what is IgG? What type of molecule is that? Okay, so this is an antibody. It's an immunoglobulin, specifically the gamma one. So immunoglobulin or antibody, same thing. So these IgGs are going to be produced from these activated B cells. So we want to know what's true. So letter A says that IgG is a divalent immunoglobulin with a variable light chain and a heavy chain. So what does divalent mean? Okay, so that means it can bind two of the antigens. It has two epitope areas, and that is true for IgG. It is divalent, and it can bind up to two antigens. It's an immunoglobulin. It's divalent. So does it have a variable chain and a light chain? So a portion of it that's variable, and it has a heavy chain, light chain. So in all immunoglobulins, there's going to be a constant portion and a variable portion. And within those variable portions, there's a heavy chain and a light chain. So that's true for all immunoglobulins, not just IgG. But specifically for IgG, it's divalent. Other ones can bond even more antigens than just two. So letter A is true. But we'll look at why the other ones are not true. So letter B says that it binds to eosinophils and targets eukaryotic parasites. So that one's not true. It's a different antibody that does that. I think it was IgE, if I remember correctly. But I might have to check that. So a different antibody does this. It might be good to know which one. Letter C says it contains a J chain. So usually a J chain would indicate that you have more structure. So it's more than divalent. So divalent, it only binds two antigens. Usually with a J chain, that immunoglobulin is going to bind more than two. It's usually around 4 or 10. Actually, it can be even up to 10, I think. So IgG does not contain a J chain. I think IgA and IgM do, if I remember from the pictures. And then produces papain. So what is this substance? What does that do? Does anyone remember what it does in terms of, yeah? So this is, yeah. So it's going to fragment it. It's going to chop it up into the FAB pieces and the FC pieces. So that constant region and those variable regions with proportional constants as well. So this is going to actually break apart IgG. It's not part of it. Or it's not going to be produced as well. So yeah, so it's not going to produce something that breaks it up. So this is a substance that breaks up IgG and other immunoglobulins. So the answer choice for this one is not none of the above. It's going to be A. It's a divalent immunoglobulin. It has variable light and variable heavy, and also that constant region. So this portion of it is true for all immunoglobulins. The divalent portion is true for IgG. Do we have questions on IgG or anything related? You okay? So number 21 is, which of the following is true about antibody production? So letter A says that during primary response, IgG is the most abundant antibody. So that's for primary response versus secondary response. So when that antigen is first introduced, actually this one is not true. So when that antigen is first introduced, IgG is not the primary response. It's not the antibody that's most abundant during primary response. It's going to be IgM. So that's why A is not true. B says that during re-exposure to the immunizing agent, T cell activation is required in order to induce memory cells to produce antibodies. So for that one, this one also isn't true because during re-exposure, it's quicker, so your immune system is acting quicker. So in order to activate T cells to induce the memory cells or to make, not even T cells, induce memory cells, in order to activate the T cells to create the antibodies is a longer process that usually happens during that primary response. So during re-exposure, that immunizing agent is actually going to activate those memory cells to just produce antibodies right away. That's why it's much quicker. So we're not going to have any T cell activation from re-exposure of an antigen that the body's already immune to or has already been introduced to. Okay, so that's why B is wrong. So A is wrong because it's not IgG that's most abundant during primary response. IgG is most abundant during secondary response. IgM is most abundant during primary. So that's primary response is when you're first introduced to that antigen, and then secondary response is that reintroduction. Okay? And then C says that memory cells from the initial antigen exposure can live up to several years. And that one is true. If it wasn't true, then vaccines wouldn't work very well. So it's asking for what is true. So number 21 is letter C. Okay. So do we have questions on antibody production, on primary response or secondary response? Okay. Okay. So then number 22 says, which of the following is an example of passive acquired immunity? So letter A says, receiving a vaccine that induces active immunity. So would that make sense for passive immunity? Okay. So it doesn't make sense because passive acquired immunity is the opposite of very active acquired immunity. So receiving a vaccine would be part of active acquired immunity, not passive. Letter B says that getting an infection or natural infection that initiates that adaptive immune response. So that's similar to the vaccine. So getting a natural infection that's going to induce that immune response is another form of active immunity or actively acquired immunity, that's not passive. So A and B are not right. And then letter C, we have cells or antibodies from an immune individual are transferred to a non-immune individual. So is that an example or is that not an example? Okay, so that is an example of passive acquired immunity. So the person isn't making their own antibodies, they're using someone else's in that case. That's kind of the hallmark difference between the two. So with a vaccine or with a natural infection, the person is making their own antibodies. And that's why it's more effective because they have those memory cells that are going to stay with them for several years. But with passive acquired immunity, the individual can't make their own antibodies, or they just don't for this case. And that's why they transfer them over from someone else who made them. And they don't last as long. Because you need that reoccurring infusion. So for 22, letter C is the answer. Okay. So do we have questions on immunity or questions on immunology, the immunology section in general? I think that's the end of what I have actually prepared. So if you guys have any questions, we can go over those. Or if you want me to go back to other ones, yeah. I don't know. I don't have access to post it on the Google sites because I'm not an SI leader myself. I'm a SARC tutor. And I don't know how I would get it online because right now this is just on my Google Docs. I don't think I'm part of the group, so I don't know if I could do that. But someone has to like accept me. I'm not actually part of the group. No, like one of the actual heads of the group have to accept me. Yeah, like there's administrators on Facebook groups. Yeah. Okay. Yeah, we can try that. I don't know exactly how I would do that. To give out my email. I could try to email people at SARC and have them post it on the Google sites, if they can do that. I'll see if they can do that later today. Yeah, you have a question? Question 5? Yeah. But other than that, remember to fill out these evaluation forms, and when you fill them out, remember to bubble in 84 at the top. It's very important that 84 is bubbled in for the session number. But other than that, I'll be here if you have any questions. Or if you want to look at this review that I put together anymore.
That was helpful, thank you.
Okay, you're welcome.
Questions like she might ask.
Yeah.
Because they're always tricky.
Yeah, and a good study tip also is to go through your notes and try to make your own questions as well. Trying to see, because you've seen her test before, and they're not going to change. It's still the same testing style. So that also helps.